So here we are back with looking at our microanatomy of a muscle cell. We talked about our two contractile proteins. Do you remember our two contractile proteins and their full I'm in trouble names? Thin actin and what was the other one? Somebody over here. Thick myosin. So what we're going to do now is we're going to look and see, okay, why is myosin thick? And when we look at the myosin thick filament, understand that component of the myofibril is composed of possibly upwards of 300 myosin molecules. And they're arranged into a cable in such a way that it looks like you've got a fairly thick log that's got a bunch of heads of Q-tips sticking up all around its periphery. Because a myosin molecule, some will describe, say, looks like a golf club. Our illustration, and when we get to a contraction, it'll make a little more sense, is your arm. From your shoulder to your fist. That's what the myosin molecule is going to look like. And so we're going to arrange it in this conglomeration of these 300 molecules with the part that's your fist able to stick up out of that thick filament. Now, for our arm, and one of these days I'm going to change that slide. For our arm, understand the myosin molecule has different subdivisions. And so from our shoulder to our elbow, that is going to be the light myromyosin, or the light chain. From our elbow to our fist, that's the heavy chain. We can also subdivide the heavy chain into S1 and S2 domains. The S1 domain of the heavy chain is your hand. The S2 domain is your forearm. The reason I like this example better than a golf club, where does a golf club bend and move? It doesn't until you break it over your knee because you shanked it into the water. But your arm bends at the wrist and it bends at the elbow. And that's what these individual myosin molecules will be able to do. So that's why I like that analogy a little bit better. So here we have a dimer of myosin molecules. We have the light chain. We have the heavy chain. There's our S2 domain. And then there you can see this globular end like your fist. That's the S1 domain. Notice, what do we have binding in the center of our S1 domain? ATP. The energy that's going to be required for a contraction to occur is going to be released in the S1 domain. So I want you to keep that in mind as we look at this organization. You see how we have this huge thick filament and all these little S1 domains? They look like Q-tip heads in our illustrations. They're sticking up all over that thick filament. That's why it looks so thick. Not only the number of molecules, but also because the S1 domains are projecting outwards. We've already talked about the flexible regions, the wrist, and the elbow of our molecules. And again, this is another way to illustrate our myosin molecules. Now, along with myosin, we're going to have proteins that are structural in nature, but also are designed to help these molecules stay in perfect alignment. Do you remember our photo we saw yesterday of the cross-section through our sarcomere and all the dots? Were you impressed with how organized they were? Think about these filaments. As the illustration showed, the little animation, those filaments are going to slide back and forth across each other. If they are not in perfect alignment, they're not going to be able to slide effectively. My daughter that just graduated high school is in the marching band. Anybody in the marching band in high school? You do the counter march and you have to walk through the other members of the band. If you're not in the right place and you happen to be coming up to the bass drum or the tuba, they're going to take you out. So that's sort of the same thing. And these structural proteins such as titin Myosin and C protein, they help hold myosin in perfect alignment and secure it in the sarcomere in the right place. And so here's our illustration. Here's our banding pattern. We have Z-disc, I-band, primarily composed of thin actin. Our A-band, portions of our A-band composed of both myosin and actin. In our illustration here, they show us the M line, and this is where we're going to have some structural proteins. But we have something that is before the M line. The M line's in the middle of this extra region that only contains myosin and not the actin. The actin hasn't extended that far. What band are we missing? The whole thing is the A band. Then the lighter region inside the A band is called the H band. So in this illustration, just this part, this overlap of the actin and myosin, would be the darkest part of the A band. Then all of this other, until we get to the M line, is going to be the H. I wish they had put that associated with this illustration. But here you see the molecule titin. It extends all the way over to our Z-disc, and it extends all the way over to our M line and binds with myosin to assist and associate with myosin and to keep it in the right place and anchor it, because myosin doesn't move. Put that in your notes somewhere. Myosin does not move. When you're looking at myosin in the center of that sarcomere, you have the two Z-discs on either side. The sarcomere is going to pull those Z-discs towards the middle. So the actin is going to slide over the myosin. The myosin is locked in place relative to that sarcomere. And so titin helps lock it in place. Now our thin filaments, they're a little bit easier to understand, because when you think of actin and our thin filament, you can really call it F-actin for filamentous actin. And filamentous actin is going to look like two strings of pearls twisted around each other. And each one of the pearls is going to represent one molecule of globular actin. So we have the actin thin filament, F-actin, a dimer wrapped around each other, and each one of the pearls is our G-actin, globular actin. Now, G-actin is important because on every G-actin molecule, there is a myosin S1 binding site. This is another reason why I like the illustration of myosin as your arm. Because that S1 domain that grabs the AT&T, it releases the energy. It is also going to be that part of the myosin molecule that reaches up off of that thick filament and grabs that position on the G-actin. If you've ever been to New York and been on the subway, you grab that handle so you don't fall down. Well, this is the handle for S1 on the globular actin molecule, and it grabs it. And when we have a contraction, even though the myosin thick filament doesn't move, that molecule of myosin grabs that binding site on G-actin and pulls the actin towards the midline of that sarcomere. And if you don't have this S1 binding site, and if myosin doesn't grab it, you'll never get a contraction. So every G-actin has that S1 binding site. So here in our illustration, here we see our two strands in purple of our filamentous actin. Each purple pearl represent a globular actin molecule, and it's each one of these then that can bind with an S1 domain of our heavy mirror myosin. Now, do you notice in this illustration there's a little piece of spaghetti that's running right through this groove of our two filamentous actin strings? And then we have these little dots that are sitting on the spaghetti. These are very critical regulatory molecules that are associated with actin. They're not so much structural. like we saw with titin and myomycin relative to myosin. But for actin, these associated molecules are first, the spaghetti is tropomyosin, which is kind of confusing, right? Because this molecule is not associated with myosin. What's it associated with? Actin. Why don't they call it tropoactin? I don't know. I didn't name it. Tropomyosin. Tropomyosin is following that groove between the two filament-actin strings, and what it effectively does is it blocks the S1 binding sites on globular actin. Whoa. If myosin has to grab actin at the S1 binding site, how does it do it with tropomyosin in the way? It can't. So when a muscle is relaxed, when a skeletal muscle is relaxed, at rest, not contracted, it's because tropomyosin is preventing the binding and preventing a contraction. So if we want to contract, we have to get S1 of myosin to the actin. So what do we have to move out of the way? Tropomyosin. We've got to move tropomyosin out of the way. And the engine that drives the movement of tropomyosin are these molecules that collectively we call troponin. Troponin are these little spheres that we saw on the spaghetti. And there are 3 of them. So the troponin molecule consists of 3 subunits, 3 component parts. And they're abbreviated TN for troponin, but TNT, TNC, and TNI. So troponin T binds to tropomyosin. Troponin I binds to actin. Where they got the I don't know. TNI is actin. So I want you to think of troponin almost like a little muscle. Because remember, skeletal muscles have an origin and they have an insertion. In this case, actin being a much bigger molecule than tropomyosin, actin is going to be like the origin. And tropomyosin is going to be like the insertion. And the key to causing troponin to move, troponin is going to exert a conformational change. It's going to change shape. And when it changes shape, it's going to be like that ratchet clamp that you use when you move. Have you ever used a ratchet clamp? When you move that ratchet clamp, it pulls on both ends. And if you've got it attached to the bed of the truck's not going to move, right? The other end is going to tighten down your load. Well, in this case, when we pull on troponin and we cause a conformational change, it pulls on tropomyosin and it pulls on actin. Actin being the bigger molecule is not going to move, and tropomyosin is pulled out of the way so that you can have a binding and then get a contraction. And what makes troponin change shape? Calcium. When calcium is released from the sarcoplasmic reticulum inside the muscle cell, and that's going to be a part of our contraction that we go through for exam 2. But in the sarcoplasmic reticulum, calcium is stored. When the action potential floods through that skeletal muscle cell, it activates voltage release channels. Calcium floods into and around all our myofibrils, binds with troponin C, causes a conformational change in shape. Myosin now can get with actin, and as long as we have ATP, we're going to start contracting the muscle until the action potential stops signaling. And when the action potential stops signaling, calcium pumps get turned on to suck all the calcium away from the myofibrils and back into the sarcoplasmic reticulum. And when we pull the calcium off of troponin C, it relaxes and goes back into its normal shape, covers over the binding sites, and the muscles relax. So calcium, not just for bones. You have to have it to release neurotransmitters. You have to have it to send the signal to the skeletal muscle cell. Now, we got to have the calcium for the muscle cell to contract. Calcium is everywhere. Calcium is super, super important. Those were functional associated proteins with actin. We also have alpha-actinin, which is a structural. It's going to anchor actin to the Z line, again, to help hold it in proper alignment, hold that spacing. And then we also have a molecule called nebulin. And nebulin, it's, for lack of a better way to describe this, it's going to wrap the entire actin filament and support the entirety of it, not in such a way to impinge upon the interaction between myosin and actin, but again, to support it, to hold it in place, and to keep it properly in line. Now, just a few more. Desmond and vimentin. These are cytoskeletal fibers. And you know we have 3 cytoskeletal elements that are defined based on their diameter. The largest diameter cytoskeletal elements are called microtubules. Then we have the smallest cytoskeletal fibers called microfilaments, and that would be like actin. And then we have cytoskeletal elements like Desmond and vimentin, whose diameters are in between the smallest and the largest. Now do you see why we call them intermediate filaments? Their diameter is in the middle. And so Desmond and vimentin, these are, and especially Desmond, vimentin to a lesser degree, but Desmond is going to be characteristic of many muscle cells. And the reason that is important, if somehow you ended up doing oncology, for instance, and you find metastatic tumors in the body, and you analyze these tumors, and you find that they are expressing the intermediate filament Desmond, what can that give you a clue about the origin, the primary tumor that those metastases came from? Desmond means muscle. And so if you have a metastatic cell that expresses Desmond, you start looking for that tumor in the muscle. And that's the key for Desmond being critical for a lot of clinical evaluations. Dystrophin is an accessory protein, not necessarily for what it does, but what happens when it gets mutated, when something goes wrong. Dystrophin, in fact, is going to help associate the myofibrils with the plasma membrane. Again, to help them to not get too far away and not too close to the plasma membrane, and to help hold them in position that way. So here we have a plasma membrane. Here we have our myofibrils. Here you can see Desmond illustrated some other accessory plasma membrane proteins. But then here is dystrophin and its association with actin. So when you hear dystrophin, have you ever heard that word before, or a word that sounds like dystrophin? Muscular dystrophy. So this was the first gene that was identified as when there is a mutation, it can lead to muscular dystrophy, the disease. Is there anything wrong with the actin? Is there anything wrong with the myosin? You just haven't adequately aligned them with the plasma membrane. But look at the debilitating disease that muscular dystrophy is, just because of a structural protein. It kind of blows my mind that sort of choreography and alignment is so critical for how muscles do their job. When we look at our banding pattern and the overlapping nature of our actin, thin actin, and thick myosin, when we get a contraction and myosin pulls on the actin, we're going to see a change to our banding pattern. The I band is going to become more narrow. It's almost going to disappear, but maybe not quite. That's why we say it narrows. The H band is going to disappear because you're filling those spaces with actin as you're sliding them closer to the M line, and now in a contracted state, the A band is going to be totally dark. And when we slide the actin and myosin, the Z line move closer and closer together. And when you have hundreds and thousands and millions of these sarcomeres lined up, adjacent to one another, a small microscopic shortening times 10 million can result in centimeters of shortening to the muscle organ. So that's how these very small molecules can result in very dynamic work that the human body can accomplish. So here is our relaxed state, our sarcomeric state that we're used to seeing. A bands, we got the H, M down the middle. And in a fully contracted state, you can see the green thin actin filaments completely fill in. So our A band is still there, and our A band is going to be the same size because myosin didn't move. M line is still going to be there. You probably can't see it because it's all dark. But now when you look surrounding the Z disc, the Z line, now we pretty much have eliminated it. It's extremely small if you're able to see it at all. And when you do that, sarcomere after sarcomere after sarcomere, that's where you can get some substantial work done.